This is the process of meiosis, but what is it? Meiosis only occurs in a few select cells, which results in half of genetic material called haploid cells. This process begins with a diploid cell that has copied its chromosomes. Meiosis 1 consists of the stages it-met: interphase, prophase 1, metaphase 1, anaphase 1, and telophase 1. Then, in meiosis 2, the process repeats except interphase is no longer included. This becomes PMAT, prophase 2, metaphase 2, anaphase 2, and telophase 2. In interphase, the chromosomes replicate, each forming a double strand that remains attached at the centromere. Next, in prophase 1, the nuclear envelope breaks down, chromosomes condense, and matching pairs of chromosomes move together and intertwine, making homologous pairs. Metaphase 1 has the crossed over homologous pairs of chromosomes move and line up randomly along the cell's equator. In anaphase 1, these chromosomes are separated and pulled to opposite sides of the poles. The last step of meiosis 1 is when the cell finishes by dividing into two new cells. This step is known as telophase 1. In meiosis 2, prophase 2 begins with the new spindle fibers forming around the chromosomes. Metaphase 2, the chromosomes line up at the equator and the spindle fibers attach to the centromeres. The centromeres divide and move to the opposite poles in anaphase 2. Lastly, telophase 2 has a nuclear envelope form around each set of chromosomes, and the two cells now divide into 4 haploid cells, all different from each other. This is effective since it helps create diversity. One way to do this is by crossing over. Crossing over is when one arm of the chromatid crosses over the arm of the other chromatid so that the sister chromatids of a homologous chromosome will no longer have identical genetic information.